Good day all. You're welcome to Wanies Academy. This is our biology class, and we want to continue with the last topic we treated, which is microscopy. So we would be able to state the importance of microscope in the field of science, and we would be able to list the functions of the component parts of a microscope. We discussed earlier that microscopy is the scientific field of using microscope to study objects. Minute, tiny objects that cannot be seen with the naked eyes, known as microorganisms, are studied using the microscope. So the microscope is a tool used to magnify or used to enlarge microscopic organisms so that the internal features, component parts, importance of such microorganisms can be seen and studied. Now, today we want to continue with the types of microscope. We have two major types of microscope. We have the simple microscope and we have the compound microscope. Microscope was invented in the 1950s, where the scientists then used lens to magnify minute objects. And from there, the knowledge of the function and role of microscope was gotten. So I said earlier that we have two types of microscope. We have the compound and the simple microscope. The simple microscope include the hand lens, which can be used to magnify images to about 6 or 7 times. It also include the light microscope, which works on the basis of sunlight, where sun rays is directed through the mirror of the microscope into the condenser to the specimen, which is viewed through the eyepiece. While the compound microscope include the electron microscope. The electron microscope is the most complex microscope. The electron microscope can magnify objects to about 4,000 times, while the simple microscope, like the light microscope, can magnify objects to about 220 times. Now, my diagram below is a very good example of the compound microscope. Now, the compound microscope generates power from light source. So the compound microscope, like the electron microscope, needs electricity to work on. So electricity produces electron, which aids the magnification of smaller or microscopic organisms into bigger form. Now, these are the component parts of a microscope. Both the simple and compound microscope have almost the same component parts. The difference is that the compound microscope or electron microscope uses electricity, while the simple microscope might not use light like hand lens, or it might use sunlight as a source to power or to view the images. Now let's start with the ocular lens. Now the ocular lens is carried by the arm. The arm where the microscope is carried as the eyepiece and the objective lens. As the eyepiece and the objective lens. The eyepiece can be worn. In the case of light microscope, it is just one, where various lenses are attached to view the specimen. Now, we have the nosepiece. The nosepiece carries the objective lenses. The objective lenses are the real magnifier. They are the tool in the microscope that enable the magnification of those tiny or microscopic specimens. So objective lens are 3. Now, depending on the size of the object or the size of magnification. In the case of the light microscope, for example, you have objective lenses that can magnify up to 5 times, up to 10 or 15 times. So the objective lenses are attached to the nosepiece. I said earlier, the eyepiece and the nosepiece are attached to what? To the arm. The arm is used to carry the microscope. Then we now move to the stage. The stage is just a platform where the specimen is placed. The specimen, for example, if, like one study, the Plasmodium. Plasmodium is a protist that causes malaria. Plasmodium is the malaria parasite. So the Plasmodium would have been prepared on a slide. We have glass slide or plastic slide. Only that the slide must allow for the passage of light, be it electron or sunlight, because without light, the images or the magnification cannot be what? Real. So the specimen are placed on the stage and they are clipped. We have the clip, two clips, which holds the specimen in both parts to prevent the specimen from what? Falling off the stage. Then after the stage, we have the condenser or diaphragm. The condenser is also adjustable. It is adjustable in the sense that we can adjust it to accommodate the amount of light or sunlight we want to pass to the object, to the specimen on the slide or on the stage. So we adjust the condenser to amount of what light that I want to focus on the specimen on the stage. After the condenser, we have the adjustment knobs. We have the coarse adjustment knob and we have the fine adjustment knob. The coarse adjustment knob brings the specimen close to the objective lens. Brings the specimen close to the objective lens, while the fine focus or fine adjustment knob gives a fine or clearer view of the specimen on the slide. Then we have the light source. Because this is an electron microscope that uses electricity, there's a light source which provides electron to view the specimen on the stage. Now, in the light microscope, here we have the lens or mirror. We have the mirror. The mirror directs light through the condenser to the specimen on the stage. The mirror collects sunlight in the light microscope, which is a simple microscope, collects sunlight and directs it through the condenser to the specimen on the stage. The importance of electron or sunlight is to view the specimen. Without light, we cannot view the specimen. So the light enables us to have a clearer view of the specimen. Then we have the stage or the base. So summarily, we have the eyepiece where the observer, the scientist or the student places their eye to view the specimen on the slide. We have the objective lenses, which is used to magnify. I say the objective lenses is of various magnification, depending on the size to which you want to magnify the object. We have the stage where the specimen is placed. We have the adjustment knobs, the coarse adjustment knob. It brings the specimen closer to the objective lens, while the fine adjustment knob gives a clearer view. We adjust it to give a clearer view of the specimen. Then we have the plane mirror in the case of the light microscope, which focuses or directs sunlight through the condenser to the specimen on the stage. Then we also have the condenser. The condenser collects sunlight or electron and directs it to the specimen on the stage. We have the nose piece or rotatory nasal piece, which houses the eyepiece, which houses the the magnification or the magnifier. We have the base, we have the clips that holds the specimen to the stage, and we have the arm or handle, which is used to carry the specimen. Now, there goes the various parts, component parts of the microscope. Till we meet next class, have a lovely day.